Good evening. Okay, any doubts from the previous class that we can take up? I hope you all can hear me. I hope you all realize that this is supposed to be an interactive doubt session. So, those of you who were not there in the previous session, or those of you who were also there, I hope you guys could access the recordings from the previous session. If there is any discussion or any doubt regarding the previous session or the anything apart from that, from the weekly assignments that you might be facing, we can discuss those things today. So last class we have completed the innate immune response mechanism, and we divided our immune system into two components, right? I'm just trying to fix something. Okay. So the event of failure of the previous recording, we have initiated the offline recording for you all, for those who have missed the live session. So we'll quickly summarize whatever we have discussed in our live class. So if you all remember, while we started our discussion with innate immunity, we divided our immunology subject as a study of immune system, and we divided it into two components, the innate and the adaptive immunity. So for today's class we will be discussing in detail the adaptive immune component. So before we move into understanding the different responses of the adaptive immune system, so for adaptive immune system, before we move on to learning the different effector responses of the adaptive immune system, we would first like to understand that what are the different features of the adaptive immune system. And as we discuss the effector responses, essentially that is how these particular features are achieved by the particular system. So if we discuss first, so the first thing is that your adaptive immune system is highly specific, and that it can distinguish between even subtle differences of amino acids, right? So even if there is, say, just one amino acid varying at a particular position, the adaptive immune system is capable of recognizing it. So if you remember from our discussion of innate immunity, we there discussed that the system is not very specific. It's not like it's absolutely non-specific, but it lacks subspecificity in terms of it will not be able to distinguish, say, in the exact the different strains of the particular bacteria. However, over here, not only strains, but just every strain of the same individual, even the subtle mutation that cause some differences between them, those are also recognized by the adaptive immune system. Apart from that, there is a tremendous diversity of receptors which enables the players of the adaptive immune system to recognize this wide diversity amongst the different microorganisms or different antigens that the body will encounter. So that is another. This amount of diversity of the receptors present on these cells of the adaptive immune system, how that is generated is what we'll read very briefly in today's lecture, but we will understand that in our subsequent classes. The third feature is that there is immunological memory, where the second encounter to a same antigen is eliciting a heightened response in a much shorter time frame, right? So we discussed that in every encounter, even with the same pathogen for innate players, is the same. So your say skin barrier is not going to respond differently if it encounters a particular bacteria for the second time. But when you are talking about adaptive immune system, the second encounter, so say if for the first encounter with the bacteria is taking 10 days time for antibody response to be generated or certain cytokines to be produced, when this particular bacteria is again encountered by the body, this 10 days time is reduced to say somewhere around 5 days time. How this memory is component is achieved is what we will discuss in today's class. The next component is that it has a capacity to distinguish between self and non-self. What that means, it is only going to recognize the antigens which are foreign to the body, right? It will never recognize anything that is present inside the host as a foreign entity and mount an immune response. Because if you can think of that situation, there are certain autoimmune disorders where your own cells of the body is attacking the other cell, leading to gradual decline in their number and overall compromise of the health of the organism, right? So there we call it a dysfunction because that is not something that is expected from the immune system. And there are certain diseases because of which such conditions are coming into effect, right? So the main point here is so it's an abnormal situation, which means the normal is your immune cells has to have a capacity to distinguish between what is the body's own cells or own antigens or what is coming from the nature, from the environment, which is foreign to the body. So these are the four features of the adaptive immune system. And we will understand one by one that how this sort of these features are achieved by the players of the immune cells, right? So while we so we'll come to this part towards the end of the discussion of today's class. So if we talk about the cells of the immune system, the adaptive immune system, there are two major players: the B lymphocytes and the T lymphocytes. So the B lymphocytes and T lymphocytes, those are like the army of the adaptive immune system, right? And they individually play a lot of role, but in cooperation with the cells of the innate immune system, these two together are required for mounting the overall response, right? So it has to be a collaborative effort between these two cells alongside the collaboration with the cells of the innate immune system. So based on which of these two cells are involved, the overall adaptive immune system again can be divided into two components: the humoral immune system or the cell mediated immune response. So for humoral, has got to do something with something present in the fluids of the body. So we will see that it's related to the antibodies which are secreted from the B cells. However, for the cell mediated component, as the name suggests, it's a cellular response where the T cells in itself are the effector molecules. We'll understand how that happens. Before we discuss in detail the effector responses, we'll try discuss it with the course of us discussing the different features of it, right? So for now, you must focus here on the B cell part. Here, so for the B cells, what is known is that they mature they form as well as they mature inside the bone marrow, right? So inside the bone marrow, while it is maturing, they express certain antigen binding receptors. So these receptors, which are when bound to the surface of the B cells, they are known as B cell receptors, which is equivalent to what we understand as the antibody molecule. We'll discuss everything about the antibody molecule, how it's the structure, how does it help in bringing about a response. But for now, I'm sure you all have heard about this particular protein, which is known as antibody molecule, and we have briefly also touched upon it during our previous lectures. So for now, though, we have an antibody molecule, which is nothing but when it is bound to the B cells, it is known as the B cell receptors. But when it is in secreted form, they are known as antibodies, which is bringing about different sort of responses. So when there happens the first antigen antibody encounter, so for the first time, when these immature B cells with certain receptors on their surface, they are capable of identifying the antigens. When this first encounter of the antigen and antibody molecule happens, these naive B cells, so naive B cells is the cells which have never encountered the antigen previously, but they are capable of uh, recognizing one. So these cells, when they're meeting, first encountering an antigen, because of clonal selection that we previously discussed, that particular antigen of a particular make is recognized by a limited population of the B cell. When this particular population of B cell is activated in presence of antigen, say, they are going to be quickly dividing and proliferating so that the number of the B cells having this particular receptor increases. How do they increase the number of receptor? By simply dividing their own cell. So as they divide, so this particular phenomena was known as clonal selection. So as these naive B cells, upon the antigen encounter, they divide. They're dividing essentially into two components. One, the plasma B cells, which is also the effector cell. So effector cell is something that is going to bring about a change, right? So these are going to produce antibodies as we were discussing. So these membrane-bound receptors are gradually going to be coming out in secreted plasma, and they can then bring about several responses. So these plasma cells, their role is to secrete out the antibodies. So they have little or almost no membrane-bound antibody. And once the infection is taken care of, their lifespan is very limited. As soon as the infection is clear, as soon as the antigenic challenge has been met, these plasma cells are going to die off, right? But what was also formed along with the plasma cells is what we call as the memory cells. Memory cells, as the name suggests, they are responsible for your secondary immune response, right? So the subsequent second encounter, that heightened immune response, the faster immune response was coming from these particular cells in the body, which are long-lived. So depending upon the severity of your initial antigen encounter, the antigenicity of those antigens, the life of the memory cells can somewhat vary, but we say that they are long lived. So in multiple years, right? So there are certain vaccinations that are given to us as a child, but they are going to protect us throughout our life. So those memory cells are say are having a lifespan of over multiple years, right? But then there are certain whenever you are getting a cut from, say, a rusted iron surface, you are suggested to take some tetanus vaccine or something like that, because there the memory response, the memory component is not as long lived, say, as the rabies vaccine that you took as a child. So the life of these B cells can vary, but to in general, we say that they are having a very, very long life. And so when these memory cells are formed, the initial nature of the initial antibody molecule, the initial selection that happened, that memory is also maintained. So these the cell receptors that are bound on the surface of these particular memory B cells, it's of the same nature as the one that has been produced during this initial infection. So it's not like we are going to show some class switching, which is present for the plasma B cells, right? Okay. So we left here where we were talking about the plasma B cells, which produce a whole lot of antibodies, and there's a particular quantification that is available where we say that each plasma cell is capable of producing more than 2000 antibody molecules per second. So even if like they live for 2 or 3 days, the number of plasma cells that are formed, the number of antibody molecules that are formed by each cell per second, it's enough for the body to take care of the foreign invasion. So because it is in secreted form, this is what is majorly compromising the humoral immune component. And when we discuss about, so production of the antibody in itself is going to bring about the effector response. This is something that is going to bring about the clearance of the pathogen. And how that can occur? That can also occur by four possible ways. One, that the binding of the antibody neutralizes the antigen, which is going to eliminate the pathogen, which means that whatever is the harmful effect that this particular antigen can bring about, say by it has some surface molecules that can bind to some other surface molecules on the cells of the host, your antibody is going to bind to them, which is going to compromise the binding ability of these antigen on your host cells. So that way it is effectively neutralizing the pathogen, the neutralizing the antigen, and the pathogen is eventually going to be cleared off. Or what it can also do is it can cross-link several antigens. So you might have certain small antigen molecules, which when bound by the antibody molecule, their size is going to increase. So this whole complex, which now has a much higher size compared to the initial size of the antigen, this whole complex is more likely to get phagocytosed, and that's how the eventual clearing, the quicker clearing of the antigen, which is now formed in clusters, is going to happen. The other possible way is that there is an activation of complement system, which is going to lyse the foreign microorganism. So complement pathway is something that is there in detail while we will understand about the. So this essentially requires the activity, the play of both the system, your innate and adaptive component, and we are going to discuss the complement system in a lot of detail in subsequent lectures. So essentially what is happening is this antibody is going to initiate a cascade of events while one event follows the other. It's multiple molecules switching to different other molecules, and eventually a toxic protein is so not at all toxic effect is produced wherein your microorganism is lysed as in there are certain pores that are formed on the cell walls of the microorganism, and that is how the rupture of the cell wall and everything is going to compromise uh, the the overall survivability of the microorganisms. Lastly, this part, and this is pretty similar, wherein we were discussing that these can neutralize the antigen, or it can also neutralize certain toxic molecules that is produced by the pathogen by coating them. So this is how the B cells are formed, how antigen uh, encounter is going to initiate a division, how the memory component and the effector components are uh, produced, and lastly, how the effector response is carried on. Over here, similarly, we have the T cells, where again, so these are produced in bone marrow, but they mature within the thymus. As they are undergoing the process of maturation, they express certain receptors, in this case called the T cell receptors, which can be of two types. One, if they have the CD4 plus make, or they have the CD8 plus make. The CD4 plus T cells are also called as the T helper cells or TH cells. So one way, so since this entire subject is very information intensive, it will be very helpful for all of you to keep on, say, identifying some small mnemonics to memorize things, right? So, a very confusing fact that I used to always find is to remember that which cell has which of the CD molecule. So, if you look at it, this 4 looks pretty much like the H, right? And if I say the CD8 positive cells are TC cells, if you place some C molecules in all possible orientation, it is going to form A. So that way, that is one way that was really helpful for me personally to remember which cell is what type. So that is one way you can remember it. But the idea is that you keep trying to memorize things, and that is the whole purpose of taking everything very slowly in detail, is for you to eventually recall everything as you go through the notes, right? Okay. So CD4 plus molecules are the T helper cells, and CD8 plus molecules present on the particular cell type, those cells are known as the TC cells or cytotoxic T cells. Okay. So we were seeing that for B cells, we have two cells forming. One of the cell is able to secrete certain molecules and it is directly interacting with the antigen. For T cells, the interaction is not very straightforward. What happens? We have a T cell which has certain receptors, say. Now, this receptor is not going to directly identify or bind the antigen. T cells require certain other molecules called as the MHC molecule or the major histocompatibility molecule. We will just look into what are MHCs and what is the function and everything in a while. For now, you must remember that in uh, alone, the T cell receptors cannot bind the antigen. It requires some sort of presentation by the MHC molecules, and only when the antigen is presented in a complex with these MHC molecules, the T cells are going to be able to recognize the antigen. For T helper cells, they only recognize antigen when they are presented by the class 2 MHC molecule. So, where are these class 2 MHC molecules? Again, these are nothing but another type of receptors which are present on all. Okay, just a second. Was some wrong information that I had mentioned. Hmm. So these class 2 MHC molecules are only present on some cells which are known as antigen presenting cells. So these molecules are not present on all cells of the body like our class 1 MHC molecules. So class 1 MHC molecules are present on all nucleated cells. So every cell of the body, every normal nucleated cell of the body is going to have class 1 MHC molecule, and any antigen which is presented in a complex with the class 1 MHC molecules are going to be recognized by the T cells, TC cells. So cytotoxic T cells, which means when they are activated because of this interaction, they are going to bring about killing function. So cytotoxic is something that is going to lead to cell death, right? So these cytotoxic T lymphocytes, they are going to eliminate the altered cells. So what are altered cells? Any cell which is having the class 1 receptor molecules present on its surface with a foreign antigen, such cells are identified as altered cells. On the contrary, if we talk about our T helper cells, which are activated upon an antigen presented to them by the class 2 MHC molecule present on a antigen presenting cell, these cells become the effector T helper cells. The primary role of the effector T helper cells is to do nothing but to release a whole lot of cytokines. So everywhere in the immune system, you're going to encounter cytokines, which are the major communication mediating molecules or in the immune system. So your cytokines can be of different types. They can be activating cytokines, so pro-inflammatory cytokines, or they can be cytokines that is going to reduce the inflammation. So anti-inflammatory cytokines. Here, these cytokines that are majorly produced, that we would just take as an example for understanding the overall response, we have TH cells, which are going to be activated into effector TH cells, which produce cytokines, which is going to activate the different cells of either adaptive immune system or the innate immune system of both the cells. Both the immune system cells are going to be stimulated in presence of certain cytokines, and these cells together are going to bring about the ultimate clearance of the pathogen. So we have understood how TC cells work, that is, they bring about the killing function. If the B cells are activated, they are going to release antibodies, which is going to take care of the infection. And if macrophages are activated, say, so their job is to phagocytose the clearance of pathogens, and that is what they are going to do upon TH cell mediated stimulation. Which type of cytokine is produced, that determines the overall response, that which particular cell is activated, or what all cells are activated, that is determined by what type of cytokine is produced. So your nature of the antigen, the nature of the interaction happening, that is going to determine the cytokine that is produced, and eventually the ultimate response is that either of the arms are going to be activated and the overall clearance is achieved. So if we talk about the T helper cells, they are the primary defense against bacteria and the protozoal cells, right? So bacterial cells, or any cell which is, say, foreign to the system, right? On the contrary, if you have any cell, any tumor cell, or say if the viruses which reside inside the host cell, that sort of infection or the tumor cells, those are going to be recognized as altered cells. So against virus and tumor cells, the CTLs are the major line of defense, right? And the CTLs is what is actually constituting the component of cellular immunity, because they are not only differentiated into the CTLs, but there's also a memory CTL that is formed, which has, again, like the memory plasma, the memory B cells, the memory TC cells also have a long lifespan, and upon subsequent infection, you will also find a secondary immune response for your cellular arm of the adaptive immune system. Okay. So just to give you a visualization of what is happening overall, so we discussed about cells, which have cell-bound antibody molecules, which are nothing but the B cell receptors, and upon activation, they might also get secreted out. For T helper cells, so this is a TCR where we have a CD4 molecule associated with this. Since it is CD4 plus, this has to be a T helper cell. And in the other case, if we have a CD8 molecule associated with the TCR, it has to be a cytotoxic T lymphocyte. And this is what we were discussing while we were talking about the MHC molecules. So if we have a virus infected cell, so this is a nucleated cell, and there might have been some virus that has infested this host cell. Certain peptides from this virus has now been presented along with class 1 MHC molecule, which is being recognized by the TC cells, and that is how these cells are going to eventually kill the infected cell, so that the infection cannot spread further. On the contrary, if we have the antigen presenting cells, they are going to express. So since antigen presenting cell is also a nucleated cell of the body, your class 1 MHC molecules are presented there as well. But if we also have the class 2 MHC molecules present on the antigen presenting cells. Any sort of, say it has phagocytosed some bacteria, and whatever antigens are associated with it, If those antigens are present along with the class 2 MHC molecule, they are recognized by the T helper cells. And based on the nature of this interaction happening here, the cytokine that is produced by the T helper cells is going to determine the downstream effect of this particular cell activation. Okay, so just a quick summary of what we have been discussing so far is, so here we have a bacteria which has entered the cell. Let's just assume that this is a bacteria for now. So whatever are the antigens present on this bacteria, inside the cell, let's just assume that this is a antigen presenting cell. We have the class 2 MHC molecules which has processed this antigen, and now they are presenting it with the class 2 MHC molecule to the T helper cell. We have an activated T helper cell that is going to produce lots of cytokines, and this cytokine is going to so th- uh, reach the B cells, the humoral component of the adaptive immune system, and ultimately they are going to uh, affect the response that the B cell have towards the antigen. Once the B cell has, in presence of these cytokines from the cellular arm, is activated, it is able to differentiate into the plasma cells, which is going to produce a lot of antibodies, and your pathogen is overall neutralized, right? Neutralized, killed, or this opsonization, whatever are the effect is best suited for the body. That is determined by what are the cytokines produced. Okay, also, if we assume that now in this case, if we say this is a virus, we have this particular invasion also processed. Just a minute. Okay, we might have to break in 10 minutes. Um, so, uh, say that this is a bacteria, and we have some uh, peptide molecules associated with this particular antigen is now presented along with the class 1 MHC molecule. This is recognized by the T C cells, and they are going to differentiate into cytotoxic T lymphocyte. This differentiation is also supported by the cytokine that are coming from the T helper cells. So that is essentially how the two arms of adaptive immune system work in coherence. Okay. Okay. So now we'll quickly see that. So we discussed how the B and T cells effectively bring about the response. But to just to put things in context, we have different receptors for your present in your adaptive immune system. We discussed about the B cell receptors of the antibody molecule, the T cell receptors, MHC class 1 and MHC class 2 molecule. We'll see all of them separately. One thing that we also discussed is the B cell can recognize the epitope alone. So epitope is nothing but, say, if you have a 2000 kilodalton protein, this is your entire protein. There must be just a, say, a small part, a stretch of, say, 10 amino acid, which is responsible for the entire antigenicity of the protein, right? So that much, that small part of the antigen is known as the epitope. So B cell can recognize the epitope alone. We have different antibodies recognizing different epitopes. However, the T cells need to have the antigen, the epitopes presented in combination with the MHC molecule. We discussed that. Okay. So before we discuss MHC molecules further, let's just see. So we now saw the players of the adaptive immune system, but now coming and we have discussed how the memory component is achieved. To now understand that how the diversity and how self and non-self tolerance is achieved, what we do, so not what we do, what our body knows that it has to do is, while this process of maturation is happening, every B cell, the gene segments that is responsible, that is encoding your antibody molecule, is undergoing a lot of random rearrangement. So there's multiple permutation and combination that is happening to bring about different gene segments together, owing to which we have uh, multiple many wide variety of antibodies which are specific to the different epitopes. So in the order of 10 raised to the power 10. So the number of antibody combinations that is actually formed is lies in this order. So these many variety of epitopes can be recognized by the B cells of your body. It is important to understand that every B cell only has one kind among all these antibody molecules, right? So in total, you have multiple B cells which which can recognize this much variety in okay. So which can recognize this wide variety of epitopes. However, every B cell, when it has matured fully, only has a single functional gene which is encoding the structural specificity of the antibody molecule, right? So every B cell can only recognize one particular kind of antigen. That is why now the cell is known as antigenically committed B cell. And during the same process, we also have something called as there's a lot of cell death that is happening, by which the B cells that by chance happen to have a combination of such a combination of gene segments that it can now recognize some component of your own body. Those B cells are selected for elimination. So during the process of maturation, what is essentially happening is every B cell is purposefully brought in contact with every antigen that is present in your body. So that is something that is happening during the course of development. Every B cell is purposefully brought about in contact with every antigen which is resident of your body, and any B cell of the B cells that is reacting to these particular antigens are eliminated by the body. So there, only the B cells, only the antigenically committed B cells that are not recognizing anything inside your body, those cells are selected to further surveil the immune system. And this is how the self tolerance is achieved. The overall process is known as selection for self tolerance, right? Similarly, if we look at the T cells, we have the bone marrow where the T cell is being produced, but it is in the thymus that the process of maturation is achieved. One thing that we need to appreciate is that unlike the B cell, we have lots of receptors present on the T cells. So somewhere around 10 raised to the power 5 receptors is present on each T cell, right? And there's also a lot of gene segment rearrangement that is happening. So similar thing is happening here also, so that your every T cell is specific to recognize particular combination of your antigen, particular complex of this antigen anti MHC molecule, right? So the order here is somewhere, and the order of diversity is somewhere around 10 to the power 9, and they also undergo selection for self-tolerance. Another way of ensuring self-tolerance is that these T cell receptors can only and only recognize antigen when they are presented as a complex with the MHC molecule. So only the antigen that is processed by the cells. So we'll just look into the endocytic and the the exogenous and endogenous processing of the antigens, and we'll see that how that is how it is helping in limiting the number of antigen that is possibly recognized by the T cell receptor. So just to understand, so this component of T cell receptors recognition only if it is of an antigen only if it is complex with the MHC molecule. When this is happening, that is also a way for the body to ensure self tolerance. Okay, so a lot of discussion we have had around the MHC molecule. It's finally our time to discuss them. So MHC molecules are nothing but it's a genetic complex with uh, multiple gene locus, right? So we have if you have some background of genetics, you would understand that it's a complete fold of DNA wherein a particular sequence of nucleotides they constitute the gene, and we can have multiple alleles, right? So when we have multiple genes placed not necessarily next to each other, say, but when it is placed as an entire complex such that multiple gene loci are present inside this complex, overall overall, this gene complex is what is encoding the MHC molecule. Okay. So, these are nothing but these are again membrane bound like proteins, which can be of two types. The class 1 MHC molecule. I don't know why I have switched everything. So, class 1 MHC molecules are present on all nucleated cells, whereas the class 2 MHC molecules are only present on the antigen presenting cell. Excuse me. Okay. Sorry, I had to plug in my laptop. So, so we have the class 1 and class 2 MHC molecules. And what is important to understand is, unlike the B cell and the T cell receptors, they do not possess fine specificity, right? So, that is something we also encountered during our discussion of innate immune system. But this is receptor of your adaptive immune system. So they do not process fine specificity, so they might not be able to distinguish between, say, the subtle differences of amino acids like the B and T cell receptors. But we will see that there is still some commonality between the epitopes that either of them is recognizing. So that we will discuss when we discuss MHC molecules in greater details. Okay, so what is important to understand is that the different alleles display a wide variation in their amino acid sequence. This variable region form a cleft. So while we're saying it's a genetic complex with multiple loci, so it can have different alleles. The different alleles, what is essentially happening is, so imagine it like is it's a structure like this one TCR, one MHC and the other MHC molecule. And based on which allele has actually encoded this particular structure, this cleft region that is forming in between, the structure, the specificity of this cleft is variable, and that is how although not very specifically, but it is able to show um, some specificity towards what kind of epitope can actually fit inside that cleft, and that is how what sort of antigens can be complexed with this MHC molecule. Okay, within which the antigenic peptides are presented to the T lymphocytes. So whatever is binding inside that cleft, that is what is going to be presented. So that is how some sort of specificity is achieved. Okay. So this overall process where a antigen is converted in a complex as MHC associated peptide fragment, this is the process which is called as antigen processing and presentation. So who is performing it? So if we were talking about, say, the class 1 binding antigens, so that is being processed in the endogenous processing system. And if it's a foreign antigen, the processing is happening uh, is what we're going to discuss as exogenous antigen processing. Okay, so we'll move one by one. Uh, we were saying that it can bind to a broad spectrum of antigenic peptides, which can be derived intracellularly or extracellular. So if we have a intracellular pathogen, say, which means that there is some foreign something present inside of the cell. So it is produced within the cell itself, such as the viral proteins or some proteins present on the cancer cell. So if there's a viral protein, what happens is this virus, it makes its home inside of the host, right? So we have a virus infecting over here. It produces the viral DNA, the viral mRNA, and ultimately a viral protein is synthesized. This protein, because every protein that has to be trafficked further, it has to enter the Golgi and the ER network. So essentially the endomembrane system. We have the class 1 MHC molecule stationed inside this endocytic compartment. Okay, this inside this endomembrane system, we have the MHC1 molecules. So these proteins, when it enters into this compartment, the class 1 MHC molecule get a chance to interact with the viral peptides. They form a complex, and instead of traveling alone, along with this MHC molecule, they travel through the trans-Golgi network and are ultimately expressed on the cell surface. So we have this viral peptide expressed in combination with the class 1 MHC molecule through this processing. So since this processing is known as the endogenous processing. So within the class MHC1 molecule, as we saw, is present inside the endoplasmic reticulum, binds the antigenic endogenous antigen fragment, and is recognized by TC cells. So that is why TC cells are called class 1 MHC restricted, because they can only recognize antigens with class 1 MHC molecules. On the other hand, we have the exogenous processing, wherein we have the antigen entering from outside the cell, say from endocytosis or phagocytosis. So we have a phagocytic cell here, and there is some antigen which is ingested inside. So something from the outside is coming inside. So it is ingested inside the phagophore. Eventually, this endosome is going to fuse with this phagophore is going to become the endosome, which will fuse with the lysosome. So here the lysosome is what has all the degradative enzymes. The eventual fusing of the endosome along with the lysosome is going to allow this antigenic peptide to be digested by the lytic enzymes present inside the lysosome. Some part of a specific peptide fragment is going to get a chance to be complex with class 2 MHC molecule, and that is how they're going to come out and get presented as a complex with the class 2 MHC molecule. So MHC class 2 molecules, which is expressed within the endocytic compartment, are complex with the peptide fragments derived from the exogenous antigen and they are now presented to the T helper cells. So the T helper cells are also called as class 2 MHC restricted. Okay, so that is a discussion about the MHC molecule. And with this, we finish learning about the receptors of the adaptive immune system. So, one thing that is important for us to also understand is it is not that where we started from, that it is not that your innate system and your adaptive system are functioning in unanimity, right? So there's a significant cooperation between the two arms that is happening, and we'll just have a quick look at how is that achieved. So we have the cells of the innate system, which so we in the innate system we also discussed that there are physical barriers and chemical barriers, but it is the cells of the innate system that plays a major role. We have the cells of the innate system expressing certain molecules, such as the pattern recognition molecules, the PRPs that we discussed. And what are others? And we also have cells which are, say, phagocytic in function, or we have cells which might have some lytic molecules, the eosinophils or the basophils, or we also have some killer cells, some cells like the cytotoxic T lymphocytes in adaptive immune arm. We have natural killer cells, which are cells of the innate immune system, which can also bring about killing function. So, once the innate immune system is, say, activated, the cellular response here uh, is is also so we also have the macrophages which can release cytokines. So we have cytokines releasing from the cells of the innate system here. These cytokines are going to activate the cells of your adaptive immune system. So we have the B cells now, which is going to produce antibodies. These antibodies can further, say, enhance the phagocytic clearance of the pathogen that we discussed. So eventually, they are going to enhance the activity of your innate immune cells, right? Similarly, we saw the T cells, the T helper cells, upon activation, they can also release cytokines. So they can be activated irrespective of the innate system, right? They can, because of some specific receptors that is present, we can have some pathogen which is activating the B cell and the T cells independently also, which is going to produce antibodies, going to help the innate immune system cells. Also, they can produce cytokines again here, which will again uh, function to support the activity of your innate immune cell. So if you see that there is a cooperation that is going on, we have the innate immune cells, and that is the first line of defense. They are taking care of the infection initially, but once the B cells and the T cells have been clonally selected and activated, they are certain cytokines produced from these cells is also augmenting, is also supporting their function, right? Individually, these cells can also function, but their activation, the molecule that is coming from the B and T cells, is also going to eventually support your innate immune cells to eliminate the pathogen. So there's just an example that we have a macrophage and microbe interaction. Some changes signals are triggered, which is going to stimulate the adaptive immune cell. This adaptive immune cell is going to further do certain things, maybe produce antibodies to neutralize or uh, cross-link the antigens, which is ultimately going to increase the phagocytic clearance of the pathogen. Also, uh, so your class 2 MHC molecule, the antigen presenting cells are nothing but the cells of your innate immune cells, right? So we have the macrophages, the monocytes, which are the antigen presenting cells in themselves. So they have the ability to stimulate your adaptive immune system. Okay, so it's just a cross link that is there. So it's just a crosstalk happening between the two systems, and eventually they are operating together to achieve the ultimate goal of eliminating your pathogen. So this is something about the memory component that we have already discussed for adaptive immune cells. So this is in context of B cell, where we have the serum antibody levels that can be measured. If you see that when the first encounter with antigen A, there is a lag period of 14 days, and we have some amount of antibody produced by that time, and the pathogen is or the infection is eventually cleared off. When there is secondary subjection of antigen A, the response comes within 6 days, and the magnitude of the response is also very, very high, right? And just for as an experimental control, along with antigen A, they, experimentally they must have also given some other antigen to see that again we have 14 days lag period for a small or a primary anti-B response. Similar experiment can also be performed for T cell response. However, it is somewhat more difficult to quantify the number of cells. So what people see, what they have done now is they put some tissue grafts, right? So for graft rejection, your T cells are primarily responsible for graft rejections because of the CTLs that can reject, that can actually damage the tissue which has been grafted in by killing the cells in it, right? So if we have a graft from some other strain transplanted in the host body, we see that the graft is rejected by 16 days time. But when the same graft is again repeated, then this time the graft rejection happens within 8 days, and as control, as different strains graft was also given, where we saw that the graft rejection happens in around 13, 14 days. So it's very similar to what we see in humoral arm of the adaptive immune system. So with this, we finish our understanding of the the adaptive, how the who are the players of the adaptive immune system, how they bring about an effect. We'll next try and understand that overall we have discussed a lot of cells in respective context, but now to put things in context of say developmental biology or differentiation, we have a common progenitor cell known as the hematopoietic stem cell, which is giving rise to all the different cells of the immune system that we have so far learned about. So there are certain markers which are present on the on this hematopoietic stem cells, which essentially help in identification of the cells, right? So how do we designate that this one particular cell is the cells of the hematopoietic, is a hematopoietic stem cell? That is why if the cell is not having something known as lin, something called as Ka is present, and something called c-kit is present. So these are certain markers for hematopoietic stem cells. You might um, come across these names if you're preparing for certain competitive exams or something. So from this one progenitor cell, we have one stem cell which is multipotent in nature. We have two progenitors that are coming about. So it can be the myeloid progenitor cell or the lymphoid progenitor cell. From the myeloid lineage, we have a dendritic cell coming in. We understand about their function. They are very important cells in the innate immune system. Or we can have the granulocyte monocyte progenitor, the eosinophil progenitor. So far we have been looking into the cells in different contexts. We understood that how each cell has a role to play, what are the effector mechanisms that they can employ to take care of the foreign antigenic challenge. So just to put things in context of developmental biology, we will now understand that how these cells are going to differentiate from a common progenitor and what all functions can, we'll just briefly discuss what all function that each one of them can play. So to begin with, all the cells that are that we have been reading about so far, they all arise from a common uh, from a common parent cell, which we call as the hematopoietic stem cell. So it's a multipotent stem cell that can give rise to all the cells of your that are present in your blood. So from this hematopoietic stem cell, we have two major progenitor cells that give rise to different lineages of cells. First, to identify the hematopoietic stem cells, it is important to understand that these are very, very rare in the bone marrow, and their number starts to increase. They begin to divide and proliferate in presence of some pathogenic infection. So, there are certain markers present on these cells to identify uh, to distinguish this particular cell from any other cell present in the bone marrow. The markers present are it should ideally be not be having a Lin marker. It should have SCA1 and CKIT. And so only the cell that is actually meeting all these three marker criteria, those are designated as the hematopoietic stem cell. In, uh, in normal developmental course, we have the cell divide into the either into the myeloid progenitor cell, from where we have the dendritic cells coming in, which is so the examples of antigen presenting cells. We'll have a quick look at all of them. So all the cells which have been marked as APCs are basically the antigen presenting cells. So we have the myeloid progenitor cell giving rise to the dendritic cell, which has the major role in being an antigen presenting cell. Apart from that, it gives rise to the granulocyte monocyte precursor, from which we have the neutrophil and monocytes coming in. The monocytes, upon antigenic challenge, would further differentiate into the macrophage, which show extremely upregulated phagocytic activity. The neutrophils, the eosinophils, and the basophils coming from the respective progenitors are named so because of particularly because of the stains that each of the granules present inside of these cells take up. So, as the name would suggest, the neutrophils are stained by neutral dyes. They have an average lifespan of 6 hours to 2 days. They have 50%, they in total comprise 50 to 70% of the white blood cells, and the granules that are present, the most active component present within them are the proteases, AMPs, and the histamines. So AMPs are the antimicrobial peptides that are, so these are all part of your the effector mechanism of your innate system. So proteases, antimicrobial peptides, all of them in presence of, so the neutrophil granules are going to be released, and these active components in contact with the microorganisms are going to clear the infection. Eosinophils, as the name suggests, so these granules take up the acidic dyes. The granules in itself is rich in cytokines, chemokines, and some cationic proteins. The average lifespan is around 5 to 12 days, and they constitute 1 to 3% of the total WBC population. Basophils are the least in number, almost it's somewhere less than 1%. They last for only up to a few hours to some days. Cytokines, histamines, and lipid mediators. So they essentially are the primary cells responsible for the innate inflammatory response that we previously discussed in our last class. Apart from that, we also have the platelets and the RBCs coming from the same progenitor, the myeloid progenitor cells. On the other hand, from the lymphoid progenitor cells, as the name suggests, we will definitely be having the lymphocytes coming in from there. So we have a T cell progenitor and a B cell progenitor, which will further differentiate into their respective lineages of producing either a T helper cell or the T cytotoxic T cell. The B cells that can eventually differentiate into a memory cell or a plasma cell. Apart from that, we again have the dendritic cells coming in from the lymphoid progenitor cell, and another cell type called the natural killer cell. So, as the name again suggests, the killer cell, which means it will have some cytotoxic function. Very much like the CTLs, which have a receptor, the T cell receptor with CD8 plus MA, these cells are also going to have the T cell receptors. Over here, the CD molecule present along with it is the CD1 molecule. So these are all certain markers that you can that are basically used for distinction of different cell types. So, among all these cells of your immune system, the antigen presenting cells that we have been discussing so far, which express the class 2 MHC molecule, we have some cells called as the professional APCs, which mean that it's their job to present the antigen along with the class 2 MHC molecule. So, among the professional APCs, we have the monocyte, macrophage, the dendritic cells, as well as the B cells, the most important being the dendritic cells. So, as a brief overview, so you would see that the processes of the dendritic cells are very elaborate. With these elaborate processes, they have a large surface area in which they can, in the entire surface area, they have the class 2 MHC molecules with the desired antigen complexed with it. The increased surface area actually enhances its chance of encountering a T cell by which it can present the antigen to multiple T cells at the same time. So these cells are the most important antigen presenting cells, and we will understand about how exactly what all they do in what different contexts with the following classes. There are also certain cells known as the non-professional antigen-presenting cells, which means that it's not their job, but under certain cases, they can also express the class 2 MHC molecule to assume the same function. So some examples of it remain the thymic epithelial cells, fibroblasts, hepatocytes, and the endothelial cells, which is present on your blood vessels. Okay. Uh, some more features into some of the cells, which is particularly of importance. So we saw that the monocytes are actually differentiating into the macrophages, and there are some key changes in its structural features that happens. One thing first, that this monocyte, again, if you have to further classify, the monocytes can also be divided into two cell population. One, which is majorly responsible for mediating this inflammatory response, and secondly, the patrolling monocytes. So patrolling monocytes are important as when so this monocyte has to differentiate into macrophage. The macrophage can either be present in circulation or it might be resident to a particular tissue, and we have different macrophages named according to the location of it. If it is a resident macrophage, so resident macrophages are actually arising from these patrolling monocytes. These patrolling monocytes, they express a lot of adhesion molecules, and they are going to slowly crawl along the blood vessels. Whenever they have a desired interaction favoring their adherence to a particular tissue, say, they're going to slow down and gradually become a resident of that particular tissue. Upon infection, the resident monocytes are now going to differentiate upon resident macrophages, and these macrophages are pretty similar to the macrophages upon in circulation, which have differentiated from the inflammatory monocytes. So, what are the structural changes that overall occurs is you would see that firstly the cell over here becomes almost 5 to 10 fold larger, and it would also have a lot of granules, especially a lot of lysozymes. We saw that the most important function that the macrophage performs is phagocytosis. So any foreign particle that is coming in, it is ingested by the cell. It is inside a structure called the phagosome. These phagosomes are going to fuse with with the endocytic compartments, where we have the endosome and the lysosome fusing together. So this foreign invader is going to be acted upon by a lot of lytic enzymes coming from the lysosome, and thereby it is going to be broken down into small um, non-pathogenic particles and eventually get released off, or they might be cleared off with certain other mechanisms. Beside that, so we discussed this. We have the neutrophils, eosinophils, different enzymes present, packed inside these granules. One thing that you will also see here is the structure of nucleus in each of these cell types. So that is another basis for classification of these cells into different types. Yeah. Mm. So the shape of the nucleus for the neutrophils, it's a multi-lobed nucleus. So that's another feature of distinction between the neutrophils. The eosinophils feature a bilobed nucleus, whereas the nucleus in basophil is almost encompassing the entire cell volume and is in shape of a horseshoe or a kidney bean shape. Okay. Apart from that, we have the dendritic cells that we touched upon. So the dendritic cells, as we were saying, is it's a very, very good antigen presenting cell. It encounters the antigen outside in the lymph node region, and it is going to capture those antigen. Once the antigen has been processed, this cell is going to present it along with its class 2 MHC molecule, and it's going to migrate out of the lymph node where uh, it is going to migrate inside of the lymph node, whereas where it is going to present the processed antigen to a naive T cell. Okay, so another thing that you must remember is that all the antigen presenting cells in itself is also expressing the class 1 MHC molecule. So if there is any alteration in any of these cells, say, taking the example of AIDS, wherein the HIV virus is actually residing inside of the Th cells. So because it's again we were discussing that for viral infection it is the cell becomes an altered cell, and that recognition is being done by the T cells utilizing the class 1 MHC molecule. So if we have any of the virus say, infecting any of the immune cells including the APCs, those cells are also going to be targeted for cytotoxic activities because they in itself are also presenting MHC1 molecules. So MHC2 molecules are only present on APCs, while MHC1 molecule is present on every nucleated cell, including the antigen presenting cells. Okay. So with this, we finish with our discussion for the innate and the adaptive immune system. We followed this with our discussion session where certain doubts regarding to the assignment and everything was taken up. In the following class, we are going to discuss the lymphoid organs, all the lymphoid organs mentioned here, and we'll get further into understanding the structure of antigen, antibody molecules, the generation of this receptor diversity of the T cells and the B cells.
Granulocytes and all of the above. So dendritic cells comes under myeloid. B lymphocytes comes under lymphoid. Then how? I think these options are not correct.
Just a second, let me open because I do not. Can you read out the entire question?
Yeah, the
Or
question is lymphoid progenitor cells do not give rise to which of the following cells?
lymphoid progenitor cell give rise to do not give
Do
rise to
not give
which?
rise to. Yeah.
Okay. Mm-hmm.
Options are dendritic cells, B
Hmm.
lymphocytes, granulocytes, and all of the above.
It should be granulocytes.
Yeah, definitely. But ma'am, then B lymphocyte comes under lymphoid, na? So how B lymphocytes is the option?
So they are asking B. The correct answer is B lymphocytes.
How can I?
Granulocytes do not come from the lymphoid progenitor cells,
Yes, yes, true, ma'am. But B
right?
lymphocyte come under lymphoid progenitor cells. No?
B lymphocytes do come under lymphoid progenitor cells, right? It's a lymphocyte. So any lymphocyte is coming from lymphoid progenitor cells.
Okay, all right, ma'am.
I think you must have got confused on what comes from it and what do not. So they ask you what doesn't come from the lymphoid origin. We have the dendritic cells coming from both the origins, right? So that is out of question. The other option is your granulocyte or B lymphocytes. So
Yes.
granulocytes are coming from the um, myeloid progenitor cells, right? So that is what is not coming from the lymphoid progenitor.
Okay, so dendritic cells comes from both the lineages.
Yes. We just
Okay,
saw.
then
Hmm.
that is what I'm confused about, sir.
Yes, dendritic cell is the only cell type that is coming from both the lineages.
Okay, okay, all right, ma'am.
Okay.
Ma'am, if the question comes, then we have to mark then we have to mark both the options or any one option.
In your weekly assignments, it's it's just one option correct questions. You cannot like select multiple options.
Okay, ma'am. And ma'am, what do we mean by lymphoblast? Is it a stage during the development of
Okay.
lymphocytes?
So lymphoblast, any cell type which has a blast word in it is a progenitor cell. Is something that is going to produce a so lymphoblast is going to produce lymphocytes.
It is an immature cell.
Right. Not exactly immature, but yes, it is going to differentiate into the type. So it's more like a more towards like a progenitor cell.
Ma'am, we can't say that it is a stem cell or something like that, sort of.
We cannot call it a stem cell because see it already. So we started from hematopoietic stem cells. So hematopoietic. So it's a very broad word. Hematopoiesis, where the formation of the blood cells start from. But here we have come down to lymphoblast. So it is only the precursor or the progenitor to your lymphocytes. That's it. It's not definitely not a stem cell, but it still has, so based on whatever is your instruction chemicals in the environment, it is going to assume the fate of a B lymphocyte or a T lymphocyte.
Okay, ma'am. And ma'am, can you tell me something about the cell addition molecule? Because in assignment question, I
Mhm.
come across this word and I doesn't know anything about that. That what does it mean and what should
Can
and
you
how
tell
should I?
me the question? Do you
Yes, ma'am.
remember?
Yes, ma'am. Ma'am, the question is this that the cell addition molecules CAMS expressed on the surface of endothelial cells are also known as. Should I also
Mhm.
dictate the
No,
option?
no. Okay, we'll hold it here. So, see, we read about, we talked about extravasation, where this particular cell has to cross the endothelium lining of the blood vessels and reach into the site of injury. Do you
Yes,
remember
ma'am.
that?
In the case of
Right?
inflammation.
Diapedesis, the marginalization. So, how a cell is getting marginalized? So, we have a lumen inside which blood is flowing, and along with that, the cells are coming in, right? For the cell to be instead of flowing, for it to now adhere to these walls, getting for it to get marginalized, there is certain interaction that is going to happen between the cell and certain adhesion molecules which is present on the walls of these capillaries. So there the adhesion molecules are going to come into play. So I think I am not sure ki there are multiple addition molecules, ICAMs, MCAMs, and so different integrin associated cell adhesion molecules. Those are called ICAMs. Now you can come to the options of it. So because we are talking about endothelium, there are certain specific cell adhesion molecules only which is going to be present on it, and some particular cell adhesion molecules on the cell which is going to get marginalized is going to be present on this particular cell. Interaction of these two adhesion molecules is what is actually slowing your cell, causing it to get marginalized, and eventually it is going to move across the endothelium. So there we have different cell interaction and different cell adhesion molecules which have a role to play. Now you can come to the entire question.
Ma'am, the options are leucine,
Mhm.
selectins, integrins, and ICAM.
So the question was what? Which cell adhesion molecules have a role to play?
Cell adhesion molecules expressed on the surface of endothelial cells are known as
Hmm.
mucin,
I think it's
selectin,
ICAM.
integrin, and ICAM.
I think it is ICAM, but I will have to check. But I hope you understand that what is the role of a cell in the adhesion molecule. So it's a we can just quickly see that which cell adhesion molecule is present on the endothelial lining, but the function of it is what you must understand. As far as I think, it is ICAMs. I'm not sure if that's correct. Maybe I should also go through the assignments before the class and I ask you to ask me a doubt.
Okay, ma'am. Ma'am, the cell addition molecule are present on the endothelial cells also and on the blood cells also.
Right. So the interaction between the two. So whenever you have two components interacting, you need something present on both of them, right? So marginalization is happening because of the interaction between the cell, the blood cell, as well as the lining of the capillary. So both of them have to interact together. So there has to be something present on both of them.
Okay, ma'am. And ma'am, we have not talked about the CD40L receptor which is present on any cells. Because we are talking about the cell receptors today, so I thought that this question.
CD40L. Okay, it's CD40L.
CD40L is present on the cells also.
That's the question. Okay, I'm not sure CD40L on which cell type is present. I can get back on that. So it is a very because I'm just sure it must be maybe some particular type of T cell is going to express that sort of receptor. But okay, in that detail we have to go. Okay, I can check that and get back. CD40L, nothing in my memory also strikes. CD40L. I'll check that and get back.
Ma'am, all the helpers on the aviation release the cytokines, but different types of cytokines
Different types of cytokines.
depending on the antigen that occurs.
Yes.
Ma'am, we have shown that if the cell is viral affected and get disease, if the cell is killed by itself by viruses, itself will be B cells. So it is a viral genetic variant, so it can get incorporated or can affect the neurocyte cells
No.
also.
So interferons is also a type of cytokine which is produced by the virus infected cell. So what is happening? We have so viruses cannot survive outside the host cell. So we have a cell which now has the virus inside of it. Once the virus infection is happening, this host cell to the host cell to the virus, along with all the class 1 MHC, along with which it is going to express the present the viral peptides and all, and eventually it is going to itself get killed. Before all of this is happening, this particular cell is going to release a chemical called interferon, which is going to tell all the nearby cells that, see, I have an infection, I have asked the immune system to kill me, but once I am dead, you have to also protect yourself from this virus to get inside of you. So there, the interferons is the communication molecule which is reaching all the nearby cells, and that is how these cells are warned against and they assume a antiviral state.
Okay, ma'am, we are now causing any mutation to the genetic level of the virus. Since
No.
I remember of the growth experiment of transforming principle, so I thought that the genetic area can come to the living cell also. It can infect those cells also, which are not viral infected cells prior.
If I understand the question right, so you're talking about that once a virus is inside the cell, so one thing that happens is this viral DNA inside the nucleus, there is a chance that this viral DNA is going to, some part of it is going to recombine and it remains inside of your this host cell DNA, right? If the cell is not killed, right, we and subsequently it is going to undergo cell division if it is a cell that can divide, and this viral DNA which is inside, so that is something that is happening in the Th cells, to the Th cells, and in the case of AIDS, right? So we have the Th cells which are constantly dying because of a viral infection. So whenever one Th cell is dying, the content of the virus is going to come out and subsequent Th cells are going to be infected. In the longer run, your all the Th cells are going to be depleted such that the overall individual is immunocompromised. But if I have a latent viral DNA incorporated into the host gene while the cell is going mitosis, for a very long time it is possible that you have multiple cell division, multiple cells which have this viral DNA, but nothing is happening. For an immune response to be triggered, remember that any sort of antigen, even if it's a nucleic acid antigen, although the antigenicity of nucleic acid is low, if it has to be recognized by the immune response and the immune response has to be triggered to eliminate this cell, it has to be presented along with the MHC molecule. If this presentation fails because the viral DNA is inside the nucleus, it has not been presented to the TC cells, this cell is going to survive. So virus, that is how the virus multiply inside the body, and unless and until there is a certain threshold it reaches, we do not get to diagnose certain things, right? So how is it able to multiply it? Because it remains in a quiescent state. In this quiescent state, there is no presentation of the viral antigen happening, so that the particular cell can be killed, or for this cell to ask the other cells to assume an antiviral state. That is not going to happen only. For an immune response triggered, you need it to come out and get present. Unless it will keep on multiplying.
Okay, ma'am. So, ma'am, if it is incorporated into the host cell nucleus, then it will be not expressed by the MHC proteins.
No. MSc presentation. So, firstly, the viral nucleic acid to be presented, as I was saying, the antigenicity of the nucleic acid. So, that will compromise our next class. Okay, where we discuss what is antigenicity. So every molecule do not have the same strength by which it can trigger an immune response. The nucleic acid antigenicity, firstly, is very poor. So if you give a macrophage and some nucleic acid particles, the chances of it eliciting an immune response against the nucleic acid is less. That's point one. Secondly, anything that is being presented along with the MHC molecule for its recognition and all, we have been so far talking about the peptide fragments, amino acid sequence. We are primarily focusing on the proteinaceous antigens. Most of the for a nucleic acid to be recognized, it first has to be in your intranuclear uh, DNA or nucleic acid antigen molecule is almost all the time failed to be recognized by the host cell, right? It either has to be in the circulation where maybe some some protein can bind to some regions of nucleic acid, but then for it has to be a very big size nucleic acid, because size also contributes to antigenicity. So those sort of things. But in terms of a latent piece of DNA or RNA inside the host genome, that being recognized and the chances of that eliciting an immune response in itself is very low. So, whenever we were discussing the entire lecture today, we were talking in terms of proteins, amino acids, peptide fragments, because protein is what primarily is recognized by most of the effector molecules of the immune system.
Okay, ma'am, got that point.
Mm-hmm. Go ahead.
No, ma'am, I got that point. There's
Okay.
something I come across in my some random thought come across in my mind
Hmm.
that fungal cell wall is also made up of chitin, so it is a carbohydrate. And since we are
Yes.
talking about here
So
peptide fragment everywhere.
after protein, the next molecule which has antigenicity is carbohydrates. But if you say give a chitin, so chitin is again a it's a polysaccharide molecule, right? So
Yes,
if
ma'am.
you give a macrophage a chitin, but then chitin also has a big size. So if I say I have the same size molecular mass protein and chitin, and I ask a macrophage to elicit a response, the response against the protein is going to be higher compared to the response against this chitin molecule, provided the two are of similar molecular weight. So these are some factors that determine antigenicity.
Okay, ma'am.
So after carbohydrates, we have lipids, and I think then comes the nucleic acid. So nucleic acid is the last in the row.
Okay, ma'am.
Also, when
And
there is a fungus, it will also have protein markers on itself. So, firstly, the patterns that we mostly talk about. Chitin itself is also a very potent immunogen, right? It can trigger a good immune response. It is also a pattern that is being recognized, but it also, with the whole organism, also has a lot of proteins on itself. So, again, proteins.
okay, ma'am. And ma'am, we know that the fungal are producing spores in some unfavorable conditions. So in that case, our body is recognizing those spores by the specific structures present on them, or how it recognize those spores, because they are made up for the unfavorable conditions, and our body for them is
Mm-hmm.
providing that conditions only. So the same immune response or will be generated by our body, or something different will be happening.
See, I think you must have heard of allergic responses, like so if there's some dust or there's some spores, there's some pollens in it. So things which are usually not mounting an immune response, we our body do not recognize it as a threat. The most likely case is these spores are going to be flushed off either from your digestive system, or maybe they are just going to be trapped in your respiratory tract and eventually flushed off by beating off the cilia or the mucus linings in there, right? So most likely it will not be triggering a immune response. But there are certain individuals who are sensitive to certain kinds of spores and certain essentially non-antigenic substances in the environment, but some individuals are sensitive to it. So something with spores, it is going to be a similar situation. So most likely they will not have any immune response that we will mount against it, but some of us might do.
Ma'am, here you are talking about the adaptive immune response or the allergic immune response. I think that you're talking about the allergic immune response that will be taking place inside the person's body. Am I
When the
right?
yes, those who are sensitive towards spores and all, it is some antibodies that is causing those allergic responses. So the part of the see, we have both the things to play. Again, these are not independent of each other. Antibody response cannot be triggered without certain chemokines triggering the activation of B cells. So your cells of the innate system, those are going to first come into effect. Certain molecules that they release, that is going to activate the B cells, which is going to produce the antibody. Once the antibody is produced, we will have the plasma cells taking care of the immediate response against, say, the spores. But we will also have memory cells now formed against the spores. So if again I am exposed to, say, the similar kind of spores, I might be in trouble. So that is an allergic response, right? But when I'm talking about the spores not causing any problem to us, we are essentially referring to the barriers, right? The gut lining, the respiratory tract lining. So there it's just the physical barriers of the innate immune system that is preventing any immune response from forming. It's simply flushing off the spores. So that's how I remain protected. But someone who is sensitive to it might actually have both arms of the immune system taking care of the spores.
Okay, ma'am. And ma'am, when our body is encountered with the antigen, then the naive B or B cell will encounter those pathogen, or the plasma cells will encounter those pathogens.
For naive B cells to become plasma cells or memory cells, what is important is an antigen encounter. So, as long as I do not have an antigen encounter, the cells are naive. Once a naive B cell of a particular specificity encounters an antigen, there you have it differentiating into plasma cells and memory cells. So memory cells is safe for the later and plasma cells is what brings about the immediate effect.
Okay, thank you so much, ma'am. My doubts are over now.
It's good that you ask. Okay, so two things I have to check. One is CD40L. I cannot still recall anything that has this receptor. It's CD, so it's definitely on the T cells, right? So sometimes you can eliminate options. So CD is something associated with the T cells.
The option is including two types of T cells, so
I
we can't
understand.
eliminate.
Okay, I'll check this and I'll also confirm the cams of endothelial lining. That's it. Okay, so next class we'll quickly have an overview of the organs of the immune system. So which cell is present in which location. I thought I'll be able to do this today, but maybe we can speed up from the next class onwards. And then we will discuss about the antigen, how one is antigenic, the other is not. And we'll try to also cover up the different types of antibody and the types of it. So for today's class, the particular image which showed all the differentiation, I'll try to enrich that image by putting in some details about the number of it, the lifespan of it on the particular image itself, and that will be uploaded as the lecture notes for you all, right? So that is all from my side today. If I miss something, I'll add on to this information in the next class. Okay.
Ma'am, currently I'm not remembering the prior session doubts, so can I ask you in the third session that we will be having on the Tuesday?
You guys are free to ask me any of the doubts in any of the classes that we meet, right? Not a problem. You can
Okay,
also post your doubts on the forum if like you do not want to wait till the next time. So over there also you will have us replying back.
yes, ma'am, sure.
Okay. Okay, I'll close the session for today. I'll see you all the next week.
Thank you so much, ma'am.
Okay, good night.
Thank you, ma'am.
Thank you.